This module describes the cryopreservation process for malus in vitro shoot tips. To start, we are removing the plant clusters from in vitro cultures and now dividing the shoots and placing them in some sterile water or liquid medium to keep them from drying out. These are 4-week-old cultures. So now we are going to show the shoot tip excision process. Once we have put the individual shoots under the microscope, we're going to peel back the leaves one by one, trying to remove the leaf and the petiole from the stem, starting at the bottom and working our way up. Once you are at the top, you can put your forceps in between each leaf and peel it back until it breaks. Trying to remove the petiole if possible. Here there's a little piece of petiole that's remaining. We're going to try to remove some of these hairs that are in the way. It's not absolutely necessary, but helpful to get a better view of the meristem area. There we're removing that final piece of petiole. And now we're down to the final couple of leaf primordia that are surrounding the meristem. There's no need to go down all the way to the meristem. Just leaving these couple of leaf primordia is fine. Now we're going to make the final cut. And here we have our finished shoot tip. That is about 1.5 millimeters. I'm going to make a few extra cuts just to trim it down a little bit smaller. And a microblade with a blade holder is very helpful for this, and some fine forceps. And there we go. There is our final finished shoot tip. And now we're going to show another example. Every shoot tip and shoot is going to be slightly different with some variability, and also from species to species and different cultivars will have some differences. There are some species and cultivars that have more of these hairs around the meristem area and on the stem and leaves. And it is always helpful to try to remove some of those to get a better view of the shoot tip area. Again, peeling back those top leaves. They break very easily when bent. Usually they will break right at the end of the petiole. And usually you don't have to do any extra cutting, but occasionally if the petiole leaves a piece of itself attached, you will need to go in there and trim it off if possible. There are some different ways to do this, and this is just an example. The main goal is just to expose the meristem area, leaving a few leaf primordia and a size of about 1.5 millimeters. It may be more of about 1 millimeter wide and about 1.5 millimeters tall. This shoot tip is slightly shorter than the last one. The remaining leaf primordia are slightly smaller. But again, it's the same idea of excising the meristem area and leaving several leaf primordia covering it. Now we're going to make that final cut from the shoot. And if you put it upright, then you can trim around the meristem area to remove any excess tissue. And making sure to get the right size. The size is very important. So you want to try to make your shoot tips very consistent so that you will have consistent results. And our finished shoot tip. And here they are plated on basil medium overnight. And then onto the pre-culture medium.